Good morning, everybody. Today,
Morning, sir.
I'm going to discuss about germplasm conservation. So, what do you mean by germplasm? The total gene, total variety, total cultivar, whether it is the wild species or cultivated species, that is the germplasm of the all the plants. It can be of rice, it can be of cereals, it can be of uh, food bearing plants, it can be of vegetables bearing plants, it can be of medicinal producing plants, medicine producing plants. So try to know what is germplasm conservation and what is the objective of germplasm conservation. So try to know what is germplasm conservation. Now you know the collection of genetic resource. What is the meaning of collection of genetic resource? Means you are going to preserve the different variety, different species, different genus of the different plant. It can be a medicinal plant, it can be of spice bearing plants, it can be of fruit bearing plants, it can be of uh, vegetable bearing plants. So you are going to maintain the gene pool. It can be, you can preserve this gene pool either in the field condition or in the lab condition. So germplasm, what is germplasm? Try to know what is germplasm. Germplasm is a living tissue from which a new plant can be grown. You know, the seed you can use as a seed, you can use the as a part of leaf of the plant, a piece of a stem, cutting, grafting, the plant tissue culture. We can use as a small section of the leaf or a small section of the stem meristematic as a explant to multiply the plants. So we are going to preserve the variety, different variety of the plants, different species of the plants. Either we multiply the plant in the field condition or we preserve that plant material inside the lab. So this is the gene pool, the total collection of all the different variety of the different plants, that is gene pool in a particular area, in a particular field, in a particular lab. Either we can using the environmental condition in the garden form or botanical garden form or inside the lab, inside the glass house, inside the greenhouse. So main motto of the objective of the germplasm conservation, that is conservation of somaclonal and gametoclonal variation in the culture. What do you mean by somaclonal? Means variation occur at somatic cell in the different variety due to multiplication of plant through plant tissue culture only with the help of chemicals and other environmental conditions inside the lab. And gametoclonal variation means maintaining the multiplication of plant with the help of germ cell. And whatever the variation come, so that should be preserved. So main motto, main objective of germplasm conservation is conservation of somaclonal and gametoclonal variation. Second objective is maintenance of recalcitrant seed, the seed that cannot germinate in all season. So you can maintain that seed for next year, next to next year. How can you maintain? At very low temperature, at a cold storage, at 1 to 9 degrees Celsius or minus 196 degrees Celsius. So there is a difference. Means what is the difference? Means cryopreservation. Then plant conservation, we can preserve the plant material at very low temperature, minus 196 degrees Celsius, inside the liquid nitrogen with the help of cryoprotectant chemicals. And second, we can store the plant part, the plant stem part, the plant root part, the plant apical meristem part, the plant leaf, new leaf portion inside the cold storage at 1 to 9 degrees Celsius. So this is the motto, just to conserve the soma-clonal variation, gametoclonal variation, then maintenance of recalcitrant seed. What do you mean by recalcitrant seed? The seed that cannot germinate in fall season. Means we cannot quickly grow that seed at a time. Means when the season will come, only then it will grow. Otherwise, it will not grow. Second, third is conservation of cell line producing medicine. Some plants that is only for production of medicine. Such type of plant that can be maintained in the gene bank. So do you know where is gene bank? National Bureau of Plant Genetic Resource. National Bureau of Plant Genetic Resource. It is present in New Delhi, Pusha. Indian Agriculture Research Institute. National Bureau of Plant Genetic Research. So in every state, the state Bureau of Plant Genetic Resource. So every state maintaining the germplasm of the different variety of the medicinal plant, cereal producing plant, vegetable producing plants, and any other. Means purpose is just to maintain the plants. Materials either in the field condition or inside the lab. So this is the main motto for the germplasm conservation is storage conservation of the cell line that producing the medicine, that producing the cereals, that producing the seeds, that producing the vegetables, that producing the fruit. Next, storage of pollen grain for enhance the longevity. You can also preserve the pollen grain of different variety of the different plants species. Means just to enhance the longevity, means we can use that pollen grain for after 2 year, after 15 year, after 20 year. So there are different methods. First is cryopreservation, and second is cold storage, and third is low pressure and low oxygen storage. There are three methods through which we can preserve the plant materials. Any part of the plant we can maintain. Okay, so we can also preserve store the pollen grain at minus 196 degrees Celsius, that is under cryopreservation. Next is delaying the process of aging. Means we can, the objective is also delay the process of aging. The objective of germplasm conservation is the delaying the process of aging. Next is, that is the storage of meristem culture for micropropagation. We have already studied the micropropagation, organogenesis, embryogenesis. It is a method for rapid multiplication of the plant through plant tissue culture. Next, conservation of plant material from endangered species. You know, the commercial variety that is not endangered species, the species that is their seed is not going to produce every year, and that is not propagated through the seed, only through a stem cutting, only through leaf. So that is going to maintain for preserving the variety, for maintaining the variety, so that it will not come in the category of endangered species, and further not come in the category of extinct, extinct. Extinct means जो विलुप्त हो गया. Next, establishment of germplasm bank. Means as per the requirement, as per the research requirement for production of hybrid seed, production, hybrid variety formation, that is essential to maintain the germplasm bank. Means we are going to maintain the variety of plant every year, so that it cannot become in the category of extinct. extinct, extinct species. Next is exchange of information and germplasm at international level. You know, basmati, it is the indigenous rice species. And earlier, America had claimed that this is the variety of our nation. But this is basically the variety of India, basmati. So they have tried to patent the basmati as टैक्सीमती, टेक्सास एरिया में भी ये ग्रो करने लगा. सबसे पहले ये बासमती राइस इंडिया में हुआ था as per the origin, but when the plant germplasm material going went to America, the Texas area, they were growing the huge quantity of that basmati, and they had tried to patent, but India had filed that this is the indigenous variety. And also the neem. So you can exchange the germplasm material, and you can make your right that this is my our plant species, our germplasm. Just like rice, there is neem also. There was also a question for neem, whether neem is the variety of India or plant of India or genus species of India or not. So Indian scientists have already made the patent against that variety, like rice, that is the basmati, then neem. So you know neem, it is the medicinal plant. And further the haldi means there is more demand in America due to corona disease. So haldi that have anti-cancerous compound, and that can use for treatment of viral disease like corona. So this is the advantage of maintaining the germ plant. So try to know germ plant. Need for the germ plant conservation. I already discussed these things. Means conservation of soma clonal and gametoclonal variation. Next, maintenance of recalcitrant seed. What do you mean by recalcitrant seed? I have already told you that that seed cannot germinate in every season. It will germinate as per the season. But some seed that can quickly germinate. Even you apply the water, the recalcitrant seed will not germinate when the season will not come. Next, conservation of rare germplasm species that used for hybridization purpose, and then the micropropagation of the plant that is have medicinal importance, and production of free virus free plant, bacterial free plant. So this is the motto for maintaining of germplasm conservation. So National Germplasm Conservation Center is in India, is in Delhi, National Bureau of Plant Genetic Resource. What is National Bureau of Plant Genetic Resources? It is present in the IRI campus, Indian Agriculture Research Institute, New Delhi. And in every state, this is the state Bureau of Genetic Resources there. Every state have such facilities. That is come to maintain the germplasm of that state. So they conserving the genetic trait means the variety that have different characters and live informed resource information resource, so that we can utilize that resource in future for our future generation for production of maximum amount of cereals, maximum amount of fruit, maximum amount of vegetables, maximum amount of medicinal compounds. So now, what is the strategy of germplasm conservation? You know, in situ, this question has come in your CBSE exam. What is difference between in situ and ex situ? So try to know what is difference between in situ and ex situ. So in situ means the maintenance, multiplication, preservation of plant in natural environment. What is the natural environment? It can be a biosphere reserve. It can be a your botanical parks. Means inside the nature, we are going to multiply that plant, not inside the lab. Means there is no artificial temperature, artificial moisture condition, artificial medium. only in the soil we are going to multiply that plant variety, plant species. In the field condition means multiplication, conservation and maintenance of the plant species in the field condition. In the biosphere reserve, biosphere reserve means there is a natural environmental condition where plant can easily multiply in suitable environmental condition, temperature, moisture, relative humidity, that is moisture, and the nutrient present in the soils. And there is a different plant species means variation of the plants. You can observe the variation like Sundarban. There is a best biosphere reserve where different variety, there are different variety of plant are there because there is a huge rainfall occurs and the soil very fertile. So this is the place where the plant is growing in natural environmental condition. Not only plant, animal can also multiply freely in free environmental conditions. So meaning of in situ means in the field condition, in the natural environmental condition. You know biosphere reserve, where biosphere reserve there is a restricted zone, and then there is a buffer zone, and then outside that buffer zone only human activity is feasible, it is permitted. You cannot do any human activity inside the core region, inside the buffer zone. Outside the buffer zone only you can do any human activity means deforestation, cultivation, like that. So plant are growing in suitable natural environmental condition in the field condition, that is known as in situ. And what do you mean by ex situ? Ex situ means protected environmental condition. Protected means artificial environmental condition. What do you mean by ex situ? The technique of conservation of all the levels of biological diversity outside their natural habitats. Natural habitats means the outside the lab, means in the forest, in the field, where there is no human activities, means you are not supplying the artificial temperature, artificial media, artificial relative humidity in case of in situ. But ex situ, you restrict that plant will grow at particular temperature, plant will grow at a particular relative humidity, that is known as ex situ. Means technique of conservation of the all level of biological diversity outside their natural habitat. Outside, I'm talking about outside that means not in the field condition, not in the forest, not in the forest area where plant are growing as per their own environmental condition. Through a technique like botanical garden, means glasshouse, greenhouse condition, and the gene bank inside the lab, means you are going to multiply, means you will supply the water as per our desire, means so that plant can multiply. You will supply the fertilizer, you will supply the manure as per our requirements. Means ex situ means under human control environmental condition, in the protected condition at particular temperature and particular relative humidity, means not in the natural condition. Now this is the examples. We can going to multiply, maintain the plants in the natural reserves, biosphere reserve, national park, marine park. So these are the examples where we are going to multiply the different variety of plants. Okay. So just see in situ means in the environmental condition, the plants are going to multiply in the environmental condition. So this is the slide diagram related to in situ conservation, means plant are growing as per the natural temperature, natural relative humidity, natural fertility of the soil, means plant can grow freely under suitable natural environmental condition as per their requirements. So these are the natural conditions where plant are going to multiply, preserve, and they no human activity for deforestation, means a strict prohibition for deforestation. Next is ex situ. Ex situ means, you try to remember, ex situ means inside the lab, inside the glass house or greenhouse means protected environmental condition. We are going to maintain the plant's parts. It can be a pollen grain, it can be a egg, it can be mm, uh, a stem part, it can be a root part, it can be a leaf part in inside the chemical. Means we are going to preserve through artificial media at very low temperature. It can be 1 to 9 degrees Celsius for cold storage, or minus 196 degrees Celsius through cryopreservation. So in case of cryopreservation, we use the chemicals. What are the chemicals? It can be sorbitol, mannitol. It can be a sorbitol, mannitol, polyethylene glycol, DMSO, dimethyl sulfoxide. So that is the cryoprotectant, a chemical that prevent the formation of ice crystal inside the living cell at very low temperature. When you put any living plant cell at very low temperature, then means at minus 196 degrees Celsius, then what will happen? There is a formation of ice crystal, and you know the equal amount of water and equal amount of ice, which have more volume. किसका सबसे ज्यादा वॉल्यूम है? अगर इक्वल अमाउंट ऑफ वाटर है, इक्वल अमाउंट ऑफ आइस है, which has more volume? कोई बताएगा? Anyone? कोई बताएगा? Which one has more volume? Yes. Are you listening my son? अनुष्का, बताइए किसका वॉल्यूम ज्यादा है? यस, बताइए किसका वॉल्यूम ज्यादा है? धीरज? Yes. So you know the volume of the volume which has more volume. The ice has more volume. So if the volume of ice is more, then it will rupture the cell membrane, and the identity of that will lost. Then the identity of that cell, the plant cell, will lost. So this is the disadvantage. That's why we use the cryoprotectant chemical. What is the name of cryoprotectant chemical? कोई बताएगा? Anyone? Dheeraj? Anushka? Can you tell me the name of cryoprotectant any chemicals? DMSO, dimethyl sulfoxide. Dimethyl dimethyl sulfoxide, it is a chemical that will maintain the uh, maintain the cell condition as natural condition. Means there no formation of ice crystal. Sorbitol, it will prevent the formation of ice crystals. So try to know what is the cryoprotectants. What is the cryoprotectant? क्या है cryoprotectant? It is the chemicals that prevent the formation of ice crystal. It is the chemicals that prevent the formation of ice crystal, that is known as cryoprotectant. That is dimethyl sulfoxide, that is the sorbitol, then mannitols, then these are the glycerols. So we add that chemicals along with the materials which you are going to preserve at minus 196 degrees Celsius. So that is very useful chemical during cryopreservation, means XC2 conservation. Means we are going to maintain that chemical at very low temperature. So that is the advantage of that XC2 conservation. So you know in this slide you will find the seed gene bank. We are going to maintain the different variety of seed at cold storage. Pollen storage, we are going to preserve the pollen grain of the different rare variety of the plants, just to enhance the longevity of that pollen, to produce the new variety. So we maintain that pollen at very low temperature, means minus 196 degrees Celsius, along with chemical cryoprotectant, that is glycerol or dimethyl sulfoxides. Next, DNA storage. You isolate the genomic DNA, and then you store that DNA at very low temperature, minus 196 degrees Celsius, for more than 20 years, not for 1 year, 2 year, 3 years. In case of cold storage, we can maintain the meristematic part of the plant, or the other part of the plant, that can be leaf part or the stem part, but it should be meristematic at a very low temperature, means 1 to 9 degrees Celsius, not more than that in case of cold storage. So there is no chance of ice crystal formation. There is no problem of ice crystal formation. There is no use of cryoprotectant in that preservation. We can also use the different method, that is low pressure and low oxygen storage. In that condition also we can going to maintain the plant materials. It can be root, it can be stem, it can be leaf. It should be meristematic, and we maintain that plant. So in vitro storage means in at lab condition we are going to maintain the different part of the plant. Okay. Now biotechnology germplasm conservation. What do you mean by biotechnology germplasm conservation? Try to know the short term storage or in vitro methods. That is, uh, means we are going to use the glycerol, we are going to use the sorbitol, or we are going to use the dimethyl sulfoxide for as a cryoprotectant, means there is no formation of ice crystal when you put that sample along with that chemicals at minus 196 degrees Celsius along with liquid nitrogen. So this is come in the category of long term storage or cryopreservation. There is other term, that is short term storage or in vitro method at low temperature, not very low temperature, at low temperature. What is the low temperature range? It should be 1 degree to 9 degrees Celsius. And in the cold storage we are going to maintain the plant part. It can be a stem, it can be a leaf, it can be a root, but it should be meristematic. We can use this for short term, means for 1 to 5 year, or 1 to 9 year, or maximum 15 year. So this is, so we can use these chemicals, but we can use the different chemicals like saline solution or glycerol, for 6 months or 1 year, 2 year, for short term storage. So try to know uh, what is the uh, what is the methods for conservation. डिस्कनेक्ट हो गया क्या? कैसे कनेक्ट करोगे बताओ? कर दो ना थोड़ा सा, बोलो। आवाज सुनाई पड़ रहा है? क्या है देखो? क्या करना है इसको बताओ? अपने आप हो जाएगा? कर दो ना तुम। हो गया? इसे बोलना पड़ेगा इन लोगों को।
की आवाज नहीं आ रही है सर बिल्कुल भी।
आवाज आ रही है? आवाज आ रही है? हाँ हाँ, ये हो तो रही है। चलो ठीक है। तो मैं ये कहना चाह रहा था कि नेटवर्क थोड़ा प्रॉब्लम चल रहा था वो, ठीक है। सो जस्ट ट्राई टू नो what is long term storage or cryopreservation, what is short term storage or cryopreservation. So try to know what are the chemical we can use for ये स्क्रीन दिख रहा है क्या? ये बताओ, स्क्रीन दिख रहा है?
यस सर।
हाँ दिख रहा है, ओके,
सर दिख रहा है।
वेरी गुड। तो ये मैं कहना चाह रहा हूँ कि लॉन्ग टर्म स्टोरेज मतलब कि ये जो दिख रहा है आपको, ये जो है लिक्विड नाइट्रोजन है, दैट इज वेरी कोल्ड, एंड यू फील द कोल्ड बर्न, कोल्ड, ठंडे से भी जलता है स्किन, try to know. So that is, we are going to maintain the plant part. It can be a pollen grain, it can be a DNA, it can be a RNA, it can be a egg, it can be a the apical portion of the leaf or apical portion of the stem, apical portion of the root. We are going to maintain at minus 196 degrees Celsius inside the liquid nitrogen. So what happens when you put any plant material inside the liquid nitrogen? It means there is a cell. This is the cell of plant part, and that cell contain the liquid. So that liquid convert into solid ice crystal. You know, the water converted, the water present in the cell of the plant will convert it into liquid, into solid ice crystal, and it will rupture. The ice crystal will rupture the plant materials. So the explant, that is that contain the cell, and that cell contain the liquid water, and that liquid water at very low temperature it will convert it into crystals. So this crystal will rupture the plant materials, the plant samples, the plant germplasm. So to prevent the identity, to prevent the such type of harm due to formation of ice crystal, we add what, cryoprotectant chemical. क्या नाम है cryoprotectant chemical का नाम? Dimethyl sulfoxide, that is dimethyl sulfoxide. So you try to know that is dimethyl sulfoxide. So this is the chemicals that is prevent the formation of ice crystal. Second, that is mannitol, that is sorbitol. So you can use any chemical, that is glycerol. So you can use any chemical so that it will not create the formation of ice crystal when you are storing any plant material at a very low temperature for more than 20 years. So whatever you are going to store, you will do the research related to that plant material after 20 years and more than 20 years. So this is the advantage of long term storage of cryopreservation. So what happens when you are going to store the any plant materials, then in that condition what will happen means you have to follow certain steps. So you know, you can observe this is the cylinder of the liquid nitrogen container, and inside that we are going to maintain the uh, any plant sample. So you are put this plant sample in that vials. Vial means जो container है उसको बोलते हैं vials, and you will put in that inside it. So this contain liquid. So if fog like a structure you are observing on the screen, that is the liquid nitrogen. That is the general nitrogen is in gaseous state, but when you are converting in the liquid state, it become very cold, and that maintain the identity of the cell along with cryoprotectant chemicals. So try to know what is the steps you follow. What is the step you follow? Selection of plant material. So selected plant material, it can be explant, it can be a pollen grain, it can be a leaf portion as explant, a stem portion as explant, and then we give the treatment of free प्री फ्रीजिंग ट्रीटमेंट पहले से, कि आपको प्रिजर्व किया जा रहा है, उससे पहले कुछ ट्रीटमेंट दिया जाएगा, उसके बाद उसके अंदर जो वाटर है उसको रिमूव किया जाएगा विद द हेल्प ऑफ प्रोसेस ऑफ डेसिकेशन. So water content of the cell is going to remove in the cell of that plant, and then vitrification means we are going to liquefy that materials, and then means we use this materials at minus 196 degree, that is known as cryopreservation, or 1 to 9 degrees Celsius through cold storage. Okay, so in case of cold storage we cannot use cryoprotectant chemical, but in case of cryopreservation we use the cryoprotectant as a dimethyl sulfoxide, ओके, और मैनिटोल और सर्बिटोल, जो मैं बोल रहा हूँ उसको याद रखिएगा अच्छे से. And then next step, when you want to use after 20 year, आप रिसर्च करना जब तक आपको नोबेल प्राइज मिल चुका रहेगा, ठीक है. So try to what you will do, you remove that plant materials from the cold storage, means preservation uh, 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 apparatus, and then what you mean thawing means converting the plant, means putting at 10 degrees Celsius, then 25 degrees Celsius, then room temperature. You cannot directly put at the room temperature, just to adjust the cell condition. जैसे कोई आता है ना, चावला, कल्पना चावला और जो गए सुनीता विलियम गई थी ये स्पेस में, तो नीचे नीचे जब इन्वायरमेंट अर्थ के इन्वायरमेंट है तो उसको अडॉप्ट करने में टाइम लगता है. सो दिस इज द प्रोसेस, मींस यू आर गोइंग टू कन्वर्ट द solid crystal materials into liquid form, that is thawing as used for the experimental purpose. Then you put the storage and then we freeze. मीन्स फर्स्ट इज वर्टिफिकेशन, क्रायो प्रोटेक्टेंट केमिकल यूज करेंगे, फ्रीजिंग करेंगे, देन स्टोर करेंगे. स्टोर करने के बाद अगर आपको यूज करना है आफ्टर 20 ईयर, देन यू पुट इन द 10 डिग्री सेल्सियस, देन रूम टेम्परेचर, नॉट डायरेक्टली एट रूम टेम्परेचर. इसको बोलते हैं थॉइंग. थॉइंग मीन्स सॉलिड से लिक्विड फॉर्म में रूम टेम्परेचर पे लाना, उसको कहते हैं थॉइंग. अदरवाइज डायरेक्टली हम रूम टेम्परेचर पे लाएंगे सॉलिड आइस क्रिस्टल फॉर्म से तो ये डिग्रेड हो जाएगा. So just to maintain the viability of the cell, just to maintain the viability of the cell, we do the process of thawing. Thawing and vertification, desiccation, it is the reverse process. Desiccation means we are going to remove the water content, okay, and we use the chemical cryoprotectant so that it will not multiply, it will not going, just it is going to dormant stage, ये मल्टीप्लाई नहीं करेगा सेल. और थॉइंग में क्या होगा कि ये आइस फॉर्म में है और लिक्विड फॉर्म में चेंज, पहले तो ये लिक्विड फॉर्म में था, आइस फॉर्म में चेंज करेगा पाउडर फॉर्म में, और देन फिर से यूज करना है तो उसको बोलते हैं थॉइंग. मींस सॉलिड फॉर्म कन्वर्टेड इनटू लिक्विड फॉर्म एंड देन वी आर एबल टू यूज फॉर एक्सपेरिमेंटल पर्पस, फॉर रिजनरेशन थ्रू ऑर्गेनोजेनेसिस, थ्रू सोमेटिक एम्ब्रियोजेनेसिस, थ्रू माइक्रो प्रोपेगेशन मेथड. तो ये फ्लो चार्ट है, पहले यहां से शुरू हुआ, then this desiccation, vertification, cryoprotectant, freezing, storage, thawing, then viability, then regeneration means multiplication of the plant. Okay, so try to know what is the multiplication. तो ये जो है आपका, आपको मैं जो बता रहा हूं ये that is the process, how can you do the preservation. Okay, मीन्स क्रायो प्रोटेक्टेंट यूज करेंगे या नहीं तो कोल्ड स्टोरेज यूज करेंगे. कोल्ड स्टोरेज का टेम्परेचर ये आपको याद रखना है, 1 से 9 डिग्री रखना है, 1 to 9 डिग्री सेल्सियस. लेकिन क्रायो प्रोटेक्टेंट का कितना रखना है, -196 डिग्री सेल्सियस. कोल्ड स्टोरेज में हम क्रायो प्रोटेक्टेंट केमिकल नहीं यूज करेंगे, क्या, डाइमिथाइल सल्फोक्साइड, मैनिटोल, सॉर्बिटोल, ये सब यूज नहीं करेंगे. लेकिन क्रायो प्रोटेक्टेंट में हम ये केमिकल यूज करेंगे. कोल्ड स्टोरेज में लिक्विड नाइट्रोजन नहीं यूज करेंगे. यू मेंटेन द टेम्परेचर ओनली 1 to 9 डिग्री सेल्सियस. इन केस ऑफ क्रायो प्रोटेक्टेंट वी यूज लिक्विड नाइट्रोजन. समझ में आया. तो थॉइंग क्या है, मीन्स यू आर गोइंग टू put the sample whatever you have stored at cryoprotectant methods in the outside condition, means you convert in from solid to liquid form. अब लिमिटेशन क्या है, लिमिटेशन ऑफ क्रायो प्रिजर्वेशन मींस जर्मप्लाज्म कंजर्वेशन क्या है, ग्रेटर स्किल चाहिए, हैंडलिंग एंड मेंटेनेंस ऑफ कल्चर रिक्वायर. मतलब कि आपको जॉब मिल सकता है इस सेंटर पे, तो आपको स्किल्ड होना चाहिए. आपको कैसे कैसे करना, सोफिस्टिकेटेड फैसिलिटी, मतलब क्या फैसिलिटी होता है, लिक्विड नाइट्रोजन कंटेनर होना चाहिए, दो टेम्परेचर कंट्रोलिंग डिवाइस होना चाहिए जहां पे हम स्टोर कर सकते हैं. सेल टिश्यू डैमेज ड्यूरिंग क्रायोप्रिजर्वेशन, ये डैमेज क्यों करता है आपको मैंने बताया है क्यों करता है. सो ड्यू टू फॉर्मेशन ऑफ आइस क्रिस्टल इनसाइड द सेल ऑफ द प्लांट. तो आइस क्रिस्टल क्यों बनेगा, अगर आपके बॉडी को डायरेक्टली -196 डिग्री पे रख दिया जाए तो क्या होगा, तो जो फॉर्मेशन ऑफ आइस इन योर बॉडी सेल, बॉडी आइस सच रहेगा लेकिन आइस बन जाएगा। तो आपको पता है कि आइस और वाटर सेम अमाउंट है, तो आइस का वॉल्यूम ज्यादा होता है, जैसे ही वॉल्यूम ज्यादा होगा इट विल एक्सर्ट प्रेशर, प्रेशर जैसे एक्सर्ट करेगा तो सेल मेम्ब्रेन रप्चर हो जाएगा, सेल का इंटीग्रिटी समाप्त हो जाएगा, सेल का जो वायबल कंडीशन में है इट विल रप्चर्ड एंड लॉस्ट इट्स आइडेंटिटी। तो इसके लिए हम कौन सा केमिकल यूज करते हैं क्रायो प्रिजर्वेशन? कोई बताएगा कौन सा केमिकल यूज करते हैं एज ए क्रायो प्रोटेक्टेंट? बताओ, एनीवन? कैन यू टेल मी द केमिकल दैट इज यूज्ड फॉर क्रायो प्रिजर्वेशन एज ए क्रायो
डाई
प्रोटेक्टेंट?
सर,
हाँ बोलो।
डाईमिथाइल
वेरी गुड।
सल्फोक्साइड,
दूसरा, दूसरा सॉर्टल,
मैनिटॉल,
मैनिटॉल, ग्लिसरॉल। तो ये आपको याद करना
ग्लाइकॉल।
है। हाँ, क्यों यूज करना है ये समझ में आ गया? क्यों समय? क्योंकि अगर सेम अमाउंट ऑफ वाटर है सेल में और सेम अमाउंट ऑफ आइस है, तो आइस का वॉल्यूम ज्यादा होगा, तो वॉल्यूम ज्यादा होगा तो ये सेल मेम्ब्रेन पे प्रेशर क्रिएट करेगा, और सेल मेम्ब्रेन पे प्रेशर क्रिएट करेगा तो सेल मेम्ब्रेन रप्चर्ड हो जाएगा, और सेल मेम्ब्रेन रप्चर हो गया तो सेल का आइडेंटिटी समाप्त हो जाएगा, सेल डेथ हो जाएगा। So that's why we use the cryoprotectant। ये बहुत बार पूछा जा चुका है मेडिकल एंट्रेंस टेस्ट में, what is cryoprotectant and how it help in make the viability of that cell at -196 degrees Celsius। What is the temperature for cold storage? कोई बताएगा what is the temperature for cold storage? कितना डिग्री रहता है
माइनस
कोल्ड स्टोरेज का? टेम्परेचर माइनस में नहीं होता है, 1 to 9 डिग्री सेल्सियस, 1 to 9 डिग्री सेल्सियस। क्रायो प्रिजर्वेशन का, क्रायो प्रिजर्वेशन का क्या कितना डिग्री होता है क्रायो प्रिजर्वेशन का? बोलो जल्दी से, टेल मी द टेम्परेचर फॉर क्रायो प्रिजर्वेशन। अभी बोला हूं -190, बोलिए माइनस, बोलोगे कि नहीं बोलोगे तुम लोग? माइनस 190।
196
ये तो आप, वेरी गुड। सो दीज आर, तो आपको बहुत ही स्किल्ड होना पड़ेगा जस्ट फॉर जर्मप्लाज्म कंजर्वेशन। सोफिस्टिकेटेड फैसिलिटी का रिक्वायरमेंट है, सेल टिश्यू का डैमेज होगा ड्यूरिंग क्रायो प्रिजर्वेशन, ये नहीं कि 100% सेल वायबल होगा, मींस वी कैन यूज द केमिकल बट सर्टेन सेल विल डैमेज। देन कॉस्ट ऑफ मेंटेनेंस ऑफ लार्ज स्केल कलेक्शन इज वेरी हाई। मतलब कि अगर आप XC2 EXSITU, व्हाट इज डिफरेंस बिटवीन XC2 एंड InSitu? कोई बता सकता है क्या है डिफरेंस XC2 और InSitu में? कोई बताएगा? XC2 मतलब क्या होता है, InSitu मतलब क्या होता है? Can you tell me what is difference between in situ conservation and EXSITU? XC2 means lab condition. We are going to maintain the plant part in the lab, either DNA or pollen grain or egg or explant of stem root inside the lab at -196 degrees Celsius or 1 to 9 degrees Celsius along with chemicals। समझ में आया? तो ये
सर,
आपको
एक
समझ में आना
C2 में
चाहिए।
ऑफसेट।
हाँ, और इन सिटू मतलब in the environmental condition, means in the field condition, in the biosphere reserve। So you are going to maintain the plant, you are going to multiply the plant along with plant materials, either the through root part or the stem part or the seed part or any part, leaf part। तो कहाँ करते हैं multiply? In the field, in the biosphere reserve, in the national park, means you are not using any artificial environment। टेम्परेचर भी मेंटेन नहीं करने की जरूरत है, न रिलेटिव ह्यूमिडिटी मेंटेन। तो ये आपको समझ में आना चाहिए। आई एम गोइंग टू शेयर दिस कंटेंट इन योर व्हाट्सएप ग्रुप एंड ट्राई टू नो, दिस इज एवरीथिंग इज रिटन इन योर CBSE टेक्स्ट बुक। ट्राई टू गो थ्रू जर्म प्लाज्म कंजर्वेशन। क्या है ऑब्जेक्टिव ऑफ जर्म प्लाज्म कंजर्वेशन? Conservation of clonal variety, gametoclonal variety, and conservation of cell line, storage of pollen grain, delay the process of aging। हो गया टाइम? टाइम हो गया बेटा? तो जस्ट ट्राई टू लर्न द दिस टॉपिक। ओके, तो फिर नेक्स्ट डे हम मिलते हैं फिर। ओके।